Good morning, and thank you for giving me the opportunity to share my work with you. And I was working on the cytoarchitectonic characterization of the lateral orbitofrontal cortex. So let's dive right in. Brodmann parcellated the human brain into several different areas based on their cytoarchitectonic structures. A region encompassing the lateral orbitofrontal cortex is since then known as Brodmann Area 47, but his work based on one single brain. Petridis adapted his labeling extended by Area 12 found in a macaque monkey in the respective region, recognizing cytoarchitectonic differences in the orbital opposed to the lateral portion of Area 4712. Years later, recent studies have identified more than one area in this region based on cytoarchitectonic characteristics. We wanted to investigate the cytoarchitecture of the lateral OFC using image analysis and a statistically reproducible approach for detecting cytoarchitectonic borders. The aim was to analyze the cytoarchitecture in a sample of 10 human post-mortem brains, to identify orbitofrontal areas in 3D space and to calculate probabilistic maps and to superimpose them with data from functional neuroimaging analysis. For this approach, we used coronally cut and histologically extended brain sections, each 20 micrometers thin, which were digitized with a 1 micrometer in-plane resolution. In a next step, contour lines and perpendicular running traverses were placed over the cortical ribbon. The gray values below these traverses allowed us to take line profiles, each represented by a 10-dimensional feature vector. For the border detection, a sliding window moved along the cortical ribbon and compared adjacent groups of profiles against each other. Each of these profiles represented cytoarchitectonic characteristics based on the volume fraction of cell bodies. Significant peaks in the Mahalanobis distance showed a structural difference between the surrounding profiles and led to a cytoarchitectonic border, revealing two dissimilar areas. The analysis was continued in all 10 brains and resulted in 232 used brain slices in which 4 different areas were identified: frontal-orbital area 4, 5, 6, and 7. Cytoarchitectonic analysis of the 4 areas showed a predominantly high cell content in the D parts of layer 3, a weak layer 4, and layer 5 being separated into 2 sublayers 5A and B. Areas FO5 and 7 revealed larger pyramidal cells in a deep layer 3, with FO7 having the larger cells and both areas being more granular, FO4 and 6 being more striate. With this procedure, 1,546 aerial borders were detected, and all delineations were manually drawn into each of the 232 used digitized brain sections. This exemplary brain is presenting a sequence of aerial distributions in the lateral OFC. Here, FO4 is occupying the anterior orbital gyrus. FO5 is found in the most anterior tip of the inferior frontal gyrus. Laterally to FO4, FO6 lies over the most part of the orbital gyrus. And finally, FO7 is to be found in the lateral orbital sulcus medially to FO6, following FO4 caudally. Based on the variable macroanatomy of the OFC, 3 different types of sulcal patterns were found in the OFC, namely type 1 with a separated medial orbital sulcus and a continuous lateral orbital sulcus, type 2 with a medial, lateral and transverse orbital sulcus assembling the typical H pattern, and type 3 with a medial and lateral orbital sulcus being divided into separate rostral and caudal portions, only being connected through the transverse orbital sulcus. Location and extent of all 4 areas varied with the changing sulcal patterns, but were still occupying the anterior, lateral and inferior frontal gyrus. Here, the type 2 pattern was found most in the extended larger sample size of 52 hemispheres. After the cytoarchitectonic mapping procedure, all 10 brains and the new areas were 3D reconstructed. In order to transfer our areas onto the MNI space, the brains and the structures were linearly registered to align them in size and position. Having all 10 brains in the same stereotactic space allowed us to integrate all 4 areas into probabilistic maps. Within these areas, the probability for each voxel being part of a specific area was color-coded. Red regions, for example, indicated that the specific area was found in this voxel in all 10 brains. Blue indicated that just 1 out of 10 brains had this areal extent. Our maps will be available in the Human Brain Project's Big Brain reference space, as well as the MNI:27 and the MNI:152 reference brain. In order to analyze the functional connectivity of the 4 new areas, we carried out a MACHM analysis. As input, we used our formally described maps in MNI:152 stereotactic space. Regions with the highest coactivation probability had been calculated and indicated as distinct cytoarchitectonic areas or macroanatomical regions. Using the SBM anatomy toolbox of Eickhoff and colleagues, revealed that the 4 lateral OFC areas show similar coactivations, with areas from the inferior parietal sulcus, the frontal pole, as well as with portions of the language processing Broca region. Additionally, unique area-specific co-activations were detected within the medial OFC, as well as the opercular, triangular, and orbital part of the inferior frontal gyrus. Functional co-activation is also occurring with the limbic system, the amygdala, in the depth of the brain, along with the hippocampus for FO7, separating it functionally from the other 3 areas. Functional analysis using the BrainMap database by Fox and colleagues revealed that Area 4 is involved in the processing of gestational input and memory. Activation in FO5 is exceptionally high in reward processing in the right hemisphere and understanding of phonology while processing language, as well as explicit and working memory shows activation in the left hemisphere. FO6 is mostly activated while comprehension of semantic context in language in the left hemisphere only, as opposed to the right hemisphere being highly activated while processing negative emotions like anger, disgust, or physical pain. Orthographic comprehension is being processed by FO7 in the left hemisphere. Gestational input and gestational and olfactory perception together with emotional evaluation of recent sensory input in the right hemisphere. These differing representations of cognitive processing and emotional evaluation are scientifically valuable because they haven't been identified in this extent until now, in the light of inter-individual variability of areal extents in stereotactic space. In conclusion, 4 new cortical areas FO4, 5, 6, and 7 were identified in a lateral OFC based on various cytoarchitectonic characteristics, suggesting differences in functionality. Macroanatomic of the OFC revealed high diversity in the arrangements of sulcal patterns, arranging the extent of the 4 new areas differently among these pattern types, with type 2 pattern being most found. The new maps will be publicly available in the HBP's Big Brain reference space, the MNI:27 and the MNI:152 reference brain. Areas FO4, 5, 6, and 7 are involved in the processing of language, sensory stimuli, working in explicit memory, and reward, adding emotional value into cognitive and sensory input as a relay station between the frontal areas and the limbic system. Thank you.